This embryology highlights screencast concerning digestive system development deals with the Foregut, Midgut, and Hindgut derivatives. The Foregut, Midgut, and Hindgut are formed when the head process and tail process develop and when lateral body folds merge ventrally. The yolk sac is continuous with the midgut. Endoderm forms the lining of the gut, and splanchnic mesoderm gives rise to the smooth muscle and connective tissue of the gut wall. This image shows an early stage and a later stage of gut development. The gut has elongated and differentially expanded into different regions. The yolk sac has been greatly reduced, becoming now just a diverticulum. The hindgut develops into a cloaca from which the allantois develops. The midgut forms an elongate loop from which the cecum develops. And the foregut exhibits a stomach expansion and outgrowths that will become the pancreas and liver. Cranial end of the foregut develops into the pharynx. The pharynx develops bilateral pouches that give rise to different structures. In particular, the first pouch becomes the auditory tube. The second pouch becomes the fossa for the palatine tonsil. This image shows further development of the alimentary tract. The pharyngeal pouches are developing. The first pharyngeal pouch is going to become the auditory tube and middle ear. The larynx, trachea, and lungs have emerged from the floor of the pharynx. The esophagus is elongating. The stomach is distending. Liver and pancreas are rising from the future duodenum. The midgut forms a loop centered at the attachment of the yolk sac. A cecum is growing out from the caudal limb of that loop. The hindgut ends in a cloaca from which the urinary bladder and allantoic stalk are emerging. The cavity of the gut is colored yellow. The lining of the cavity is formed by endoderm, and splanchnic mesoderm surrounding the endoderm becomes smooth muscle and connective tissue, except that in the region of the pharynx and most of the esophagus, myotomes migrate to the surrounding splanchnic mesoderm, and the musculature there is skeletal muscle rather than smooth muscle for the pharynx, the larynx, and most of the esophagus. Both the pharynx and the cloaca end blindly. They're closed by a membrane that is formed internally by endoderm and externally by ectoderm. These membranes then have to break down as a stomadeum grows outward to form the mouth, and a proctodeum grows caudally to extend the anal canal slightly. The stomach arises when a segment of foregut expands dorsally to form the future greater curvature. The stomach rotates to the left so the dorsal surface is facing left, and it assumes a transverse position when the liver displaces the cranial end. Increased growth along the cranial edge of the greater curvature produces a fundus of the stomach. In ruminants, a complex stomach is formed with 4 compartments. The compartments arise embryologically as diverticula of a simple stomach. In this image, we see transverse sections of a developing stomach. These are cranial views, so left is to your right. At an early stage, we see dorsal and ventral mesenteries, with the liver developing in the ventral mesentery. These would be mesogastrium, a dorsal mesogastrium and a ventral mesogastrium. At a later stage, we see the dorsal border facing to the left. The ventral mesogastrium is divided into a less secure and a falciform ligament by the growing liver. The dorsal mesogastrium has a spleen developing in it and also a lobe of the pancreas. This is the future greater omentum. On the right, we see the adult pattern, a ventral view with the stomach aligned in a transverse plane within the abdomen. The complex ruminant stomach has 3 fermentation compartments: the rumen, which has dorsal and ventral sacs, the reticulum, and the omasum. The abomasum is a glandular compartment analogous to the simple stomach seen in carnivores. Embryologically, the fermentation compartments arise as outgrowths of an embryonic simple stomach. The rumen and reticulum arise as outgrowths from the fundus region. The omasum arises as an outgrowth of the lesser curvature. Initially, the midgut is anchored by the yolk sac. As it elongates, it actually herniates through the umbilicus, temporarily protruding outside of the embryo. As the yolk sac atrophies, midgut, lacking a ventral mesentery, is free to rotate, and it rotates 360 degrees around the future cranial mesenteric artery. This creates the caudal-duodenal flexure and the transverse colon. A jejunal diverticulum is a developmental anomaly produced by a persistent yolk sac. Another developmental anomaly is a atresia or stenosis of the gut. During development, the gut temporarily fills up with epithelial proliferation, and it must recanalize. If in certain locations that recanalization fails, you have an atresia or a stenosis, a narrowing of the lumen. This image shows midgut rotation around the future cranial mesenteric artery. Here's the start, here it's halfway around, and here rotation is completed. The rotation produces a root of the mesentery around the duodenum and colon loop. This image shows a developmental anomaly, a jejunal diverticulum that can be a source of colic. It represents a persistent yolk sac that was not absorbed. The diverticulum could be a fistula, where it continues through the abdomen, or it could be reduced to a fibrous cord. In either case, it can be a source of colic. In ungulate herbivores, the large intestines undergoes modification to form a spiral colon or a large colon. All of the ungulate modifications involve the ascending region of the colon. The transverse and descending regions are always present, but this ascending region initially forms a loop, and that loop develops into a spiral colon for the pig, a coil colon or spiral colon for ruminants, and a large loop that fills the ventral abdomen and even is incorporated in the proximal cecum in the case of the horse. The embryonic cloaca, which is closed by a cloacal membrane caudally, is divided by a septum into a ventral, urinary, and genital chamber and a dorsal region which becomes the rectum and anal canal. Most of the anal canal is lined by endoderm, but the anal canal is extended caudally by an ectodermal outgrowth called a proctodeum. The proctodeum produces a cutaneous zone from which anal sacs arise in carnivores. Between the endoderm mucosal region and the ectoderm cutaneous region, there is an anal membrane. The anal membrane is the dorsal part of the cloacal membrane that was separated by the urorectal septum. That anal membrane must degenerate for defecation to be possible in the neonatal animal. This image shows earlier and later stages of a cloaca closed by a cloacal membrane and being divided by a urorectal septum into a ventral and a dorsal region. Here we see that the ventral region will become the urinary bladder and genital system, and the dorsal region will become the rectum and the anal canal, closed by an anal membrane that must degenerate so fecal matter can pass through. And the anal canal is extended by a proctodeum that provides a cutaneous zone caudal to where the anal membrane is located. An hepatic diverticulum, which grows outward from the future major duodenal papilla, gives rise to the liver, the gallbladder, and one lobe of the pancreas and the pancreatic duct. The other lobe of the pancreas and the accessory pancreatic duct originate as an outgrowth of the minor duodenal papilla. Depending on the species, one of the pancreatic ducts will be larger than the other. In the dog, the accessory pancreatic duct is larger than the pancreatic duct. In the cat, the accessory pancreatic duct is small and usually is not even present. This image shows the accessory pancreatic duct arising from the future minor duodenal papilla and the hepatic diverticulum arising from the future major duodenal papilla. The hepatic diverticulum gives rise to hepatic ducts and lobes of the liver, to the cystic duct and gallbladder, and then becomes the bile duct, as you can see over here in the adult pattern. Also, the hepatic diverticulum gives rise to one lobe of the pancreas and the pancreatic duct, which comes off near the origin of the hepatic diverticulum. This image shows the pancreas developing as two lobes. As development proceeds, the two lobes cross one another and anastomose. This creates a single pancreas with a body and two lobes, and the duct systems within the pancreas anastomose. So the pancreas can be drained by two ducts. Depending on the species, one of the ducts will be larger than the other. Mesenteries are formed by splanchnic mesoderm at the same time that the gut is formed. We recognize a dorsal mesentery and a ventral mesentery. The dorsal mesentery is complete, and in the adult, regions of mesentery are named for the portion of gut that they attach to. So we have a mesorectum, a mesocolon, a mesoileum, a mesojejunum, which together form the mesentery, a mesoduodenum, and a dorsal mesogastrium, which becomes the greater omentum in the adult. The spleen develops in the dorsal mesogastrium. Ventrally, a mesentery is absent in the midgut region because initially that was occupied by yolk sac, and that was the region of gut that elongated and herniated through the umbilicus. Caudally, the ventral mesentery of the cloaca becomes median ligament of the urinary bladder. Cranially, liver develops in the ventral mesogastrium, dividing it into a lesser omentum and a falciform ligament. This image shows that a root of the mesentery is formed as the gut rotates 360 degrees around the future cranial mesenteric artery. Also, the mesentery, the mesojejunum, mesoileum, greatly expands as the jejunum and ileum elongate. This concludes this embryology highlights screencast dealing with development of the foregut, midgut, and hindgut.